to the Division of Education, Innovation and Energy e-learning program. I am Ms. Jamilla Amin Bacchus, facilitator for CAPE Biology, Units 1 and 2. Today we continue our lesson, Unit 1 Biology, Sexual Reproduction in Plants. So let's go. We are going to be looking at pollen germination, fertilization, and plant embryogenesis. You would have recalled we looked at this very picture, and I know many of you were really impressed with the mangoes. Mangoes are in season now, so I know we will want to have an idea as to how and what is the reason for the process of sexual reproduction. And as we said here, what is the significance of double fertilization? Why do plants carry out double fertilization? Animals don't. We don't have double fertilization in animals. You simply have one fertilization for the meeting of the male and the female gamete. But in plants, we have this happening twice. We have double fertilization. What is the reason for this? So let's look at what you need to know going into this lesson. What should be your previous knowledge? You should have an appreciation of the flower structure, a dicot flower structure. You should appreciate and understand clearly the steps involved in mitosis. We should have also done pollen, grain, and ovule formation and structure. And we should have had a good appreciation of pollination. What is pollination? The different types of pollination. What are the specific objectives? What would I like you to know at the end of this lesson? There are three things again. I would like you to know certain things. I would like you to have certain skills to be able to do certain things. And I would like you to be able to appreciate certain things after doing this topic. So cognitively, we'd like you to be able to explain the process of pollen germination, to outline the steps involved in double fertilization in plants, to justify, to say why this process of double fertilization in plants is necessary, and to outline the steps involved in embryogenesis in plants. That is, how do you form an embryo? How do you go after fertilization to the formation of an embryo in plants? In terms of the psychomotor, we would like you to be able to draw, label, and annotate how the pollen grain germinates. We'd like you to be able to draw. Now, these things are important because for CXC, for your SBAs, you're required to be able to draw these structures. Also, why are we doing this? At the end of the day, what do we want you to appreciate? Why do you need to appreciate? Why do you need to do the topic of sexual reproduction? Why is it important that you understand double fertilization? We would like you to know that, and we'd like you to be able to answer exam type questions. So here are some exam type questions. These were actually taken off some past paper questions. So again, get your phone, take a picture of it. While we are going through this exercise, I would like you to attempt to answer it before we come to the end. Right? So while we are going through, you are going to get some information, and I would like you to be able to attempt it. But these are the type of questions that you can be presented with in a CXC paper 2 question paper. Right? So first one, outline the sequence of events that leads from germination of a pollen grain to double fertilization in flowering plants. And secondly, briefly comment on the significance of double fertilization in the life cycle of a flowering plant. These are just two. There are many other questions, but what I would like to to do with you as we look at these questions is not just to look at the content, but to understand how the questions are posed and what are the key parts of the question you need to appreciate going in to be able to maximize your responses and your marks that you can achieve in doing these questions. Good. This is another question, right? So again, get your phone, take a picture of it, and attempt it, and at the end we'll come back to it and we will answer it together. Okay, you can see a drawing here and some questions that are based on the drawing. Good? I'm giving you some time to make sure and get the picture just right. In our last lesson, we would have looked at the topic of the formation of the male and the female gamete in plants. We would have looked at the stages that you go through to form the pollen grains and to form the ovule. So I'm just recalling this because we would have seen these two diagrams in our last lesson, but you need to have the appreciation of them going forward into this lesson today. Okay, so again, recall the structure of the pollen grain. You know that you have two nuclei inside of the pollen grain. In the female gamete, you are seeing this. Now, this is a frequent mistake that students make. They have a lot of challenges with understanding the structure. Bear in mind, the actual gamete is this part inside of here. Bear in mind, the actual gamete is this part here. And very often, a lot of students feel this entire thing here is the gamete. Okay? So please be careful with that. This actually is the gamete. And you can see this gamete actually has 8 nuclei inside of it. In the final analysis, you are going to have you will have 3 cells here, each having a single nucleus, and 3 here, these cells here, this one as we mentioned last day, these cells here are the synergids. This cell would be the female egg cell. These 3 here are the antipodal cells, and in the middle here we have 2 nuclei which are called the polar nuclei. They will eventually fuse. So inside of the female gamete, you actually have 8 nuclei, and they would have been produced, as we saw on the last occasion, through the process of mitosis. And this is very important at CAPE level for you to appreciate this. At Form 5 level, it was not necessarily compulsory at CSEC level, but at CAPE level, it is very important that you understand the structure of this structure here, which is the embryo sac, and precisely how you get the 8 nuclei form. Good? So let's move on from here. Good. So I'm just recalling for you, I'm just showing you the structure of the ovary again, because this is where a lot is going to be happening. If you recall, these here would have been you can look at it, the anther. You seen the filament attached to it? So this is an anther, and I know it looks very big, but it is the anther, and on top of here you would have the stigma, and here you have the ovary with the ovules, and inside of the ovules you will have the embryo sac. So the embryo sac is the gamete, and that is inside of the ovules. Good. So I want you to do something for me, because I know you may have been reading up previously, and I want you to attempt to draw the pollen grain going through germination, the germination of a pollen grain. Can you do that for me? I'm going to give you 2 minutes to do it, and then we're going to come back. So we'll take a little break here. In the meantime, see if you can draw it on your own without looking at the internet or in textbook. See if you can draw the pollen grain germinating.
Go ahead.
Okay, welcome back. So I would have given you an assignment to draw the germination of the pollen grain. Let me see if I can do it, but we have some slides there. I'll show you them later, but let me also attempt. Since I'm giving you the assignment, let me see if I can do it. So if we have the pollen grain, normally we have the okay, so we have the stigma, right? And we will have the style. So we have the stigma, the style, and the ovary. So the pollen grain, when it lands on the stigma, initially it has the generative nucleus and the pollen germ tube nucleus. And what happens is that you begin to have the pollen germ tube nucleus directing the formation of the pollen germ tube. So we would have had the two nuclei inside of this pollen grain. And as it begins to germinate, you would have had one of the nucleus, the pollen tube nucleus, actually direct the formation of the germ tube. So this will move downwards and it will push the formation of this germ tube all the way down the style. So what you would have seen is that there were two nuclei. One is solely responsible for the formation of the germ tube, and the other one that contains the male gamete. And what happens now is that this will further carry out the process of mitosis and form two cells. So you'll have two nuclei and they will actually form two cells. So this one will be no more because this would have gone through the process. And so at this stage, a drawing like this will show the process of germination of the pollen grain. But if they ask you to do a drawing showing the germination of the pollen grain, you should have seen the germ tube nucleus at the front and the two nuclei from the generative nucleus following down. This is the drawing more or less. Not exactly my best work, but it can give you a good idea as to what you should be showing if they ask to see a diagram or a drawing showing the germination of the pollen tube. So this simply outlines the steps involved in the pollen grain germination. The ovules are contained within a hollow organ of the pistil called the ovary. The pollen is deposited on the pistil's receptor surface, the stigma. On the stigma, the germination of the pollen grains begins by absorption of water and nutrients, and the pollen grain produces a tiny pollen tube through the style of the ovary. So as I indicated in the drawing before, initially when the pollen grain lands on the stigma first of all has to be receptive. So it has to be of the same species, and once it is of the same species, once it has met all the criteria in terms of, encouraging cross-pollination and so on, you will find that this, the stigma will now allow this pollen grain to, form its pollen tube. But what directs the formation of this pollen tube is the pollen tube nucleus. And that will cause the formation. Simply, if we looked at the structure of the pollen grain, you will see there are some pits within the membrane of the pollen grain, and it is through those pits that you will find the pollen tube will grow out and begin to grow down through the style. So the tube nucleus enlarges and comes out of the pollen grain through one of the germ pores to form a pollen tube. The tube nucleus descends to the tip of the pollen tube. Good? So the tube nucleus is at the tip because it is leading the formation of this tube. The generator cell also passes into it. It soon divides into 2 male gametes. Further on, in an act of double fertilization, one of these 2 sperm cells within the pollen tube will fuse with the egg cell of the ovule, and that will become the new embryo. The second sperm cell will in fact form the endosperm. It will fuse with the pollen nucleus and become the endosperm. Good? So this is simply stating that the other nucleus combines with the 2 subsidiary sexual nuclei of the ovule, which initiates formation of a reserve food tissue. I hope you are appreciating this. It initiates formation. It initiates formation of a reserve food tissue. That's the crux of it there. Why double fertilization? Because this new organism will need to have a food source. And so this food source is the endosperm, and in some plants it is the cotyledon. The endosperm can actually help with the formation of the cotyledon. So that's the crux of it here. Why double fertilization? Because this new organism needs to have a solid food supply. It cannot go out there depending on its environment. It needs to work with its food supply for its growth into the new, its new life. Good. So this again illustrates what we attempted to do. It's showing the transition. This would have been the pollen grain, and you saw the structure of the pollen grain, which you should know by now. And then you saw where from the 1 nucleus, you had the 2 nucleus form. You remember that happened within the anther. Even before it emerged out of the anther, this occurred. And then you would have had, so you had the nucleus, the generative nucleus, and you had a cell wall forming around the nucleus because it is a plant cell. And eventually, what would have landed on the stigma would have been a structure such as this. And this here shows the pollen germ tube growing down. Let me just show you it in another direction. Good. So you may be able to appreciate this a little better because the germ tube grows down. You're going to see the 2 male nuclei, they are following the tube nucleus. So the tube nucleus is directing the whole show. So as I attempted to do in my drawing, you're seeing here a better drawing of this germination. So this is a drawing of the germination of a pollen germ tube. Good. So while we are looking at what was happening at the top of the flower, at the stigma, here we have in the ovary development taking place. So this would have shown us the structure of the embryo sac. And as we can see here, we are seeing the 3 nuclei on this point. We are seeing the other 3 nuclei here, and we are seeing the 2 in the middle, as I pointed out earlier. Now, I am going to ask if you can recognize and identify which 3 or which one will be the egg. Well, it's identified here. But if it was not identified, how may you have known that that was the egg cell? Normally, the egg cell is the largest cell inside of the embryo sac. It's the largest one. And it will have the 2 other nuclei, which are the synergids, and on the opposite end you have the 3 antipodal cells. Now normally what you will observe is that the egg cell is closest to the micropyle. So wherever the micropyle is, you will see the egg cell within the triploid structure of cells there. So if you want to figure out which end would be the antipodal cells and which end would be the egg cell, look to see where the micropyle is. Because in some books you may see the antipodal cells on one end, and in other books you'll see on the other end. So people get confused because they say, Miss, I don't understand. Look for the micropyle. And the 3 cells that are closest to the micropyle would be the 3 that include the 2 synergids and the 1 egg cell. Okay? So this is a beautiful picture of what is happening inside of the ovary. You can actually see the cell structure. So this was taken with a microscope, and you can actually see what is happening here. It's very, very beautiful. Again, this is showing now double fertilization. Here, this would have been the pollen tube that would have come from the top. So this would have more or less been extended out to go up to the top of the stigma. And this tube, as you can see here, it is bringing down the 2 sperm cells. You should have had the pollen germ tube cell at the front, but that will eventually break down. Once it's finished its job of forming the pollen germ tube, it has no other role to play, so it eventually will break down. These 2 sperm cells, each of which is haploid, they will now do their work. One of them will merge with the egg cell. So one will merge with the egg cell, and this is your zygote. This is your new plant, and it is 2N. The other nucleus will merge with the polar nucleus that is in the middle of the embryo sac, and this now will form your endosperm. Since the polar nuclei was diploid and you had 1 cell joining which was haploid, this endosperm is now 3N, it is triploid. Good? So the endosperm is triploid and the zygote is diploid, and you have this structure. This eventually will develop into what we call the seed. So this entire structure will now be known as the seed. Initially it would have started off as the ovule, and eventually it becomes the seed. Here we're seeing it again. So here you can see the 2 sperm cells, and you have the germ tube cell that is ahead. The nucleus is here. The purpose of this one was simply to form this tube. It was directing the formation of the tube. So once that tube is completed its job, this no longer has any part to play in this process. It will eventually break down. These 2, however, these are the sperm cells, the male gametes, and what will happen is that one will join with the egg cell, and that will become the new zygote. That is the new plant for the next generation. The second cell will join with the cell that is in the middle, which was the polar nucleus, and this become a triploid structure, because remember, this central cell would have had 2 nucleus initially forming this polar nuclei, and you would have had the cell now joining with it, so it's now triploid, and this would become the endosperm, which is a food source. Let me just say something about the endosperm, because the endosperm, if you look at a dicot cell, you will not, a dicot plant, sorry, you would not necessarily see a large endosperm. The endosperm actually breaks down in the dicot seed, and it actually, a lot of the material, a lot of the energy that it would have had actually goes on to be taken up by the cotyledon. And so you will you will not see a very large endosperm in a dicot plant. In a monocot plant, however, like corn, maize, you will actually see a pretty large endosperm still there. But the endosperm is initially the cell that will be formed when you have the second fertilization. So here is where we are seeing double fertilization, because you had, remember, the fertilization is the merging, the fusion of the nucleus of the male gamete with the nucleus of the female gamete. So you had a fertilization occurring here, and you also had a fertilization occurring here. And this is what we call double fertilization occurring. You had two fertilization occurring. Now, what is the reason for that? Again, as we pointed out earlier, since you need to have this plant continuing on its own, the seed is not carried by its mother. It's not carried by the parent tree. It is going off on its own. And so it needs to have a food source, so that when it is planted in the ground, it can survive until it can make its own food. We all know the process of making its own food is photosynthesis, correct? So until that plant gets above ground and it can photosynthesize, something has to supply it with its energy. And here is where the endosperm or the cotyledon come in. They will supply it with energy. Here, I just wanted to point out to you that some ovaries have more than one embryo sac. Some ovaries have more than one ovule. You can see corn here. And one of the ways you can identify that you have more than one ovule, you're seeing all these little strands here that we all know when we peel corn, we have these problems with all these strands to remove. If you look carefully, you will see that every one of these structures here has a strand leading to it. This strand is actually a pollen germ tube. So that every corn seed that you have there has to have a pollen grain go down that tube and actually merge with the female nucleus. Interesting, isn't it? So next time you're peeling that corn, you're going to look closely and you're going to see, right? So the ones at the end, some of them, you may see them flattened, is because you may not have had fertilization occurring with them and so on. And even sometimes between the corn, you'll see some parts flattened. And, you know, that is why you will find, in Trinidad, I know, there are lots of people who plant corn and they plant it close together. The closer you plant it, the chances of the pollen grain landing on the entire landing on every one of the stigmas to encourage fertilization increases. So when you see people planting corn, one here and the other one 6 feet away, you understand that maybe they don't understand how this process of fertilization occurs. And that's why sometimes the corn looks very, very scanty. You find one corn seed here and the other corn seed there. So this is quite interesting. Just take a look at it and you'll see you can actually follow each pollen tube to every single corn seed. Each corn seed has its own pollen tube. Interesting, isn't it? Good? So the last thing we're going to look at today would be embryogenesis, plant embryogenesis. After fertilization occurs, what happens next? Good. You have to now go from one cell, zygote. The zygote, remember, is simply the cell where you had the fused male nucleus and female nucleus. They fuse, that is called a zygote. How do we go from that to a matured embryo? And this just shows the development of this embryo. This is a diagram. This is a series of diagrams that you are supposed to have a good working knowledge of. You're supposed to be able to follow the stages in the development of the mature embryo. So this is the final product. Good? Now, you can get a lot more information on this online and so on, but it's not really too important at this stage that you go into great depth. But you need to have an appreciation of the development. So you start off with the fertilized egg, which is what we would have had when you had the egg cell merging with the male gamete and the nucleus of the male gamete. And then from this, mitosis takes place. Remember that process? Mitosis takes place, and therefore we had two cells formed. One cell is called the apical cell, and the other one the basal cell. That basal cell, take a look at it. It's actually what is going to keep that whole structure attached. That's the basal cell. And then you have the octant. What do you think octant means? Octant, 8, right? 8. So you have 8 cells within that structure, and then you have the globular, and then you have the triangular, and this part here is called the suspensor. You have the heart shape, you have the torpedo shape. By here, you are seeing the cotyledons shaping up nicely. Inside of here is where you have the vascular tissue developing. So in this area here, you're seeing the vascular tissue, right? In this area here, you're seeing the vascular tissue. We know the xylem and phloem would be in that area. And to the front here is where you have the meristematic tissue. The shoot apical meristem is in this area. That is what will form the first leaves of the plant, because these are the cotyledon. Cotyledons are not leaves. They were the remains from the storage organ. And to the base down here, you have the root structure. The root apical is in this area, and that is going to give rise to the roots on this area here. So basically, when you are finished with the process of embryogenesis, you have in the seed a plant that is ready for germination. You have something that if you put into the ground, right, once it has the right conditions, it can now grow up to become a matured plant. So we would have seen that we went from a flower, good, and eventually we go to a seed, and from the seed we can have a matured plant. Good. After fertilization, in angiosperms, you know what angiosperms are. Those are modern day plants. So you have dicots and monocots. Those are angiosperms. You know, the plant kingdom is divided up, and the last or the most matured plants are the angiosperms, the dicots and the monocots. In angiosperms, the ovule consists of 2 layers. The outer layer of 2 protective coverings called the integuments, and the inner cells that form the nucellus. After fertilization, the ovule matures into the seed, and the integuments form the outer seed coat of the seed. Of course, we know that is the tester. In dicots, the seed coat consists of 2 distinct coverings, a typical firm outer layer called the tester, and a generally thin membranous inner coat called the tegment. The nucellus may disappear completely or may exist as a thin layer called the perisperm, and in some species, right, the ovary wall becomes the fruit. The ovary wall becomes the fruit. Remember, the fruit's role is simply to help to disperse the seed. The important part here is the seed, because the seed contains the next generation. The fruit is simply a tool that the plant is using to entice animals or to be carried by the wind or so to be able to help in dispersing the seed. The seed has the next generation, the fruit does not. That is why we can eat a mango and the fruit is going to disappear. But the seed is what we have, and you toss the seed over the way, and pretty soon you get a tree from there. The fruit really doesn't go into the next generation. The seed does. Good? So let's get back to our past paper questions. Basically, we would have gone through the process of the germination of a pollen grain, the steps involved in the formation, in the process of double fertilization, and in embryogenesis in plants. So I'm certain you can answer the questions now. How did you do? Okay, so the questions were: outline the sequence of events that leads from germination of a pollen grain to double fertilization in flowering plants. Good? So you were supposed to outline, right? You were supposed to literally go in a sequential way, list the events which occur from germination of a pollen grain, from the time the pollen grain lands on the stigma, and you have the stigma yielding to the pollen grain. You are supposed to outline what is happening then until double fertilization. So if we go back to some of what we saw earlier, what can you tell us is happening? What is happening there? Firstly, we have the pollen germ tube nucleus is going to initiate the formation of the pollen tube. It leads the way in this pollen tube formation. Then we have the generative nucleus carrying out mitosis and beginning to follow the pollen germ tube nucleus down into the pollen tube. They make their way all the way down to the micropyle, and the pollen germ tube nucleus will disintegrate, and the two nuclei that remain, eventually the first one will merge with the egg nucleus in the female gamete and become the new zygote, and the second nucleus will merge with the diploid nucleus and become the endosperm, which is the food store. So that is the sequence of events that leads from germination to double fertilization. So once you outline all those steps, you're going to get your 5 marks. Again, briefly comment on the significance of double fertilization in the life cycle of a flowering plant. Again, why is it important for double fertilization? Why not single fertilization? Well, if we had single fertilization and we had the zygote formed, then when the seed is planted with the zygote and there's no food source, the zygote may die. And that is one of the reasons why, if you have a seed and it is a very small seed, let's say like a tomato seed, and you put that too far down in the ground, you plant it too deep in the ground, you will find it will not emerge, because the tomato seed obviously has much less food storage than, let's say, a corn seed. So you realize you can plant a corn deeper than you can plant a tomato or lettuce. Normally, if you're planting a lettuce, you will just put the seed on the top and just sprinkle some soil just above it. You cannot dig a hole and put it in. You just sprinkle some soil above it because there isn't a lot of food source. So obviously, for this plant, for this new embryo to develop, to grow up, it has to get food. And the food source we have is the endosperm. And so double fertilization ensures that no matter where this new plant goes, no matter where the seed is deposited, it has walked with its food supply. It don't have to depend on its environment to get its food supply. It has its ready-made food supply. And so the chances of survival of this organism, of this species, is going to be increased. Good? That's the main reason for double fertilization. That's the benefit to double fertilization to the species. Good? And here we have the drawing again. So you would have seen the micropyle is in this area. So we know the egg cell would be one of these 3 cells, usually the largest one, right? The middle cell would be the polar nucleus, and these here, the antipodal cells, these eventually would disintegrate, and these will become the endosperm. So what you are supposed to do here, comment on the fate of the structure label X. So the structure label X, what is the structure label X, people? If you look at it, this here is the stigma, right? This here is the style, this whole region here is the style, and this here is the ovary. But once fertilization occurs, this ovary now becomes the fruit, right? So this area here is the fruit wall, and this fruit wall is important because remember, look at our mango that we saw earlier on. You were attracted to the mango. You wanted to eat that mango. You see people on Facebook with their mangoes and people want the mangoes because the fruit wall helps to disperse the seed. So the fate of the fruit wall is that it becomes the fruit, and it may attract insects to it, it may attract animals to it by its structure, and eventually it will lead to help with the dispersal of the seed. And they also ask, what comment on the fate of Y? The fate meaning what eventually would happen with it. Comment on the fate. What will eventually happen with this? So Y is what? If you notice, Y is this area in here. And if you look at it closely, this is the seed. Once fertilization has occurred, this is now the seed. What is the fate of the seed? The seed eventually will give rise to a new plant. The seed is carrying the young plant, and it is also carrying the food reserves, and its fate is to ensure the survival of the species. It is going to hopefully find a suitable environment in which it will deposit itself, and once it gets the right environment, it will grow, and it will ensure that this new organism is going to be able to be successful. The advantage of this whole thing is that sexual reproduction is occurring. Meiosis has occurred, and so what you have now is a unique organism. It is genetically unique in that because meiosis has occurred to form the gametes, there's no other plant like this plant. It's unique, it has a unique genetic makeup, and so it increases the chances of this species continuing. It increases the chances of this species surviving in spite of environmental changes. That is the advantage of sexual reproduction as opposed to asexual reproduction. Okay, so at this point, we're just recapping the specific objectives we had. Explain the process of pollen germination, outline the steps involved in double fertilization in plants, justify the process, why is double fertilization in plants important, outline the steps involved in embryogenesis in plants. As I said, this is a very short area. We don't expand too much on it at this level, but you need to have a solid appreciation of what is happening during the formation of the embryo. You should also know how to draw, label, and annotate. And I want you to continue this beyond this session here. Draw, label, and annotate the pollen germination. So when you label it, make sure you do your necessary justified labeling lines. You know what your labels should be, either small caps or script lettering, and you do that for the pollen germination, and also to draw, label, and annotate the sequential steps in double fertilization. So like we showed in the diagram before, you are supposed to show the steps involved in the process of double fertilization. So this is the pollen germ tube that would have made its way here, and the pollen grain, one nucleus would have merged with this and the other would have merged with this. So you are supposed to be able to make a drawing of this diagram and show what is taking place during double fertilization. And lastly, you're supposed to answer your exam type questions and have a good appreciation of this topic. I trust that you would have had a deeper appreciation of the process of sexual reproduction in plants. We started off looking at the structure of the flower and the important parts of the flower that are involved in sexual reproduction. We looked at the process of pollination and why the need for cross-pollination as opposed to self-pollination. We also would have followed up here with the germination of the pollen grains, the process of double fertilization, and embryogenesis. So you can see this plant, in order for this plant to emerge, this seed had to have had something in it to keep it growing, to provide it with energy so that it could have emerged and it could have continued to carry out the metabolic processes so that it can grow up to now be able to photosynthesize as it gets above ground. And we have our beautiful mango tree here, and of course we have our mango fruit that we all are enjoying at this time with all different types of desserts and likewise. Good. So I trust that this session would have helped you to get a better appreciation and be able to feel a little more secure in your knowledge going into exams. I will be following up with a new topic in our next session. We will be looking at sexual reproduction in humans, and I know that is something that you will love to look forward to. So see you in the next class. Have a good day. Bye.